Hello and welcome to week 3 of biotechnology. This week we're going to deepen our understanding of bacteria and viruses and learn more about the tools that are needed to manipulate these microorganisms and also to identify them in the laboratory setting. The essential questions we're going to ask this week are how bacteria and viruses can be used to develop technology. What tools do we have to identify, transfer, and grow genes within these microorganisms? How the Human Genome Project and HeLa cells impacted the field of biotech? Some potential benefits, ethical concerns surrounding CRISPR technology? How drugs interfere with the disease pathway? And how we can test the efficacy of proposed drugs using the scientific method. Our live session this week is a duplicate session, and it's meant to prepare you for one of your summative assessments, the Pharmaceutical Development Project. This live session is called Clinical Trials, in which we'll develop a drug and test that drug using the scientific method to make sure that it has its intended purpose. Some other key assignments are to complete a virtual lab to identify the technologies needed to identify microorganisms, participate in discussion of ethical issues surrounding CRISPR, and to create a prototype of a drug needed to treat a human disease. As always, feel free to use the weekly agenda and pacing guide to make sure that you're completing assignments in a timely manner. Use the weekly checklist to make sure that you've completed all the work you needed to complete, and email me with any questions or concerns. Thank you.